So I can see a few charts, something on Bunya virus, herpes, rabies, HPV, herpes, Onyong, HIV mutations, HPV, heterogena, rhabdoviruses, herpes, Onyong. Okay. If you could be more specific, I would be happier. What specifically did you learn about the topic? Like if it's rabies or herpes or Edward Jenner. In the meantime, a clinical presentation of rabies, hydrophobia, aerophobia, chickenpox, is herpes viridae and not pox viridae. HPV causes cervical cancer. That is true. Different ways of classifying viruses. Yeah, so I can see you've picked quite a number of things. I will now talk about influenza viruses. And today I would like you to answer in the chat. I think it will be faster. You have finally gotten to your clinical years, or you are seeing some patients, and this 24-year-old female presents with a running nose, headache, malaise, and sore throat. So which of the following is the most likely cause? Which one of these would you finger as the most likely cause of this condition? This is what you would call an upper respiratory tract infection. If you are to pick one of these viruses, of course, this is a lot about epidemiology on what is common. You still need to do a diagnostic test. But if you are to test this, say one by one, which one would you pick? So somebody is talking about C rhinovirus. Everybody seems to know that rhinovirus is the most likely cause, and you're all right. Rhinovirus is the commonest cause of common colds or upper respiratory tract infection. So you're right. And the reason for this question is so that you can appreciate that even though influenza is very important, it is one of the leading causes of death across the world. It's not the leading cause of upper respiratory tract infections. So you still need to consider other pathogens. And so when you're looking at a patient with a sore throat, with a running nose, with a cough, think about all the pathogens, including bacterial ones, like that can also cause sore throat, for instance. So think globally. Don't just think about, oh, because you learned about influenza, this is the only thing that can cause this kind of disease. It is not. So today we'll talk about the taxonomy or classification of influenza. We'll talk about the epidemiology, the structure, how it replicates, the antigenic variation. And this is probably a new concept in terms of what you've learned so far in virology, antigenic variation. And it's an important one. We usually emphasize a lot on it in influenza because it determines a lot of things about influenza. We've also seen it with the COVID-19. So they are similar, but not the same. And I encourage you to be very keen on antigenic variation to see how it happens. And this is a concept we'll carry on for the rest of your medical career. Pathogenesis, as we've done in the past, clinical features, how to diagnose influenza, and treatment and prevention. I'm looking for my pointer. The influenza as a disease has actually been around for thousands of years. It has been mentioned as far back as 400 BC by Aristotle. It has been documented in the Middle Ages, but people really didn't know what it was called, what was causing it. And the outbreaks were happening, but people could not identify because we were not able to identify viruses until early in the 20th century. Actually, not early, but yeah, we could say in the first half of the 20th century. And the statement that influenza is caused by influenza viruses looks like a dumb statement, but it needs to be considered in the background that a lot of people talk about flu. I have a flu. So when somebody says they have a flu, are they referring to influenza, or influenza-like illness. And a lot of times we are referring to influenza-like illness. And there are many other causes of this kind of illness, this kind of syndrome. Of course, if there is influenza going around, and you see somebody with an influenza-like illness, which is basically fever, sore throat, upper respiratory tract infection symptoms, they are most likely having influenza. But there could be other causes which still need to be ruled out if influenza is not the cause. It tends to be seasonal in the northern parts of the world, and basically the subtropics, the Europe, most parts of the US and Australia and other parts in the south. But I think in Kenya and in other parts in the tropics, it is throughout the year. So it circulates throughout the year because we have sort of, our weather doesn't change much. And although in the West, winters are the worst with influenza, in Kenya, we don't see this kind of seasonality. We might see peaks in terms of the numbers, but you can get influenza anytime during the year. I'm sure recently you heard about an outbreak reported in Nakuru. Transmission usually occurs in low temperature and with low humidity. And this is because during winter, for example, as a classic example, it's very cold, but also very dry. All the water in the atmosphere has been, all the moisture in the atmosphere has been turned into ice. So it's very dry. And so when you cough, for example, droplets find very little resistance. They will travel further and they can infect more people. And this virus, influenza, and several other respiratory viruses prefer to replicate in lower temperatures, say at 33 degrees Celsius, as compared to the body temperature, which is 37. So during winter, when somebody is inhaling cold air, the upper respiratory tract will be cooler than the rest of the body. So it forms the right atmosphere for these viruses to replicate and then contiguously spread downwards into the lower respiratory tract and cause problems. So this low temperature and low humidity favors transmission of influenza in winter. The term flu that is loosely associated with these symptoms should be used carefully. So as you talk about influenza or flu, be sure of what the patient is referring to and what you're talking about. They're not synonymous. What is the global burden of influenza? Lower respiratory tract infections are some of the leading causes of death anywhere in the world, but especially in sub-Saharan Africa. Influenza causes death across the world, and in fact, most deaths might be outside sub-Saharan Africa. And a lot of these are due to lower respiratory tract infection. When influenza actually causes viral pneumonia, or it worsens an existing situation, say a chronic lung disease in the elderly especially, or also in the very young. But usually in the elderly, people with heart or lung disease tend to be the most hit. And so in a year, we get about 500,000 deaths, but the number of severe cases are up to 5 million, although the infections are many more. The reason we don't report all infections is because most of them will not go to hospital. The only severe cases will go to hospital. And influenza can be very severe, even in young, healthy guys like you, and you end up going to hospital. In fact, it's one of the leading causes of job losses due to what are they called? Off days. So many companies lose a lot of that. Some of the companies in Kenya give their workers influenza vaccines to try and reduce this. In terms of risk, the circulation in winter in the north, you can see here is usually November right now to April next year. January, I think, is the coldest, so tends to be the worst. In the tropics, circulation is all year, throughout the year. In the south, you can see South Africa, Australia, and parts of South America. The worst period is from April to November. So they're just coming out of their season. They are now getting into their warm season, and so they will not have as much. And for this reason, even the vaccine recommendations vary from the northern hemisphere to the southern hemisphere. And you'll find that whatever is circulating right now is what you want to vaccinate against in the northern hemisphere. But this might change so that when, by the time it's April or February next year, the southern hemisphere will need a different set of vaccine combinations so as to be effective. And we'll discuss that a little later. And that's why the influenza vaccine is given every year to try and cater for this circulation, because whatever is circulating now might change. And that seasonality is one of the reasons that we need to discuss antigenic variation. This year is award for influenza patients, I think in 1918. If you remember your history, the First World War was ending in, I think, 1919. Influenza at that time, this was called the Spanish flu. At the time, Spain was non-aligned, so they were not having a lot of propaganda in terms of the influenza. Other countries refused to report on the influenza because they wanted to keep the morale high because there was war. But Spain continued to report on the influenza, so it was called the Spanish flu. But it was actually across the world, and there was a lot of movement of people and troops. And because of that, a lot of people got infected across the world. Influenza itself, this pandemic caused more deaths than the war. In fact, it's estimated to have cost between 25 million and 50 million deaths. The war cost, I think, around 20 million. So this is, I think, probably the most well-known outbreak of influenza. And it was recorded because of the number of deaths and the impact that it caused. Taxonomy is straightforward, actually, for influenza because we have 4 genera, and each genera has 1 species. And you can see here the family is called Orthomyxoviridae. Orthomyxoviridae. There's a related class which you should not confuse, the family called Paramyxoviridae, which includes measles and other similar viruses. We will discuss that in due course. So this is Orthomyxoviridae. And Orthomyxoviridae has 4 has other species, other viruses that we may not talk about, but the ones that we are most interested in are these ones that actually cause influenza. And even when we talk about influenza, most of the time we're referring to the first 2 genera, alpha influenza virus and beta influenza virus. C doesn't cause severe disease, and D has only been isolated in other animals like cattle, not in humans. And you can see here, if you talk about alpha influenza virus as a genus, the species is influenza A virus. For beta influenza virus genus, the specific virus is influenza B virus. So a lot of times we'll be talking about influenza A and influenza B. The rest, not so much. In terms of Baltimore classification, which as you know has 7 groups, this is group 5, and it's negative sense, single-stranded RNA virus. If you remember, our last topic, I think, was on rabies or rhabdoviruses. They had a similar genome, except today, the influenza virus genome is segmented. The rhabdovirus genome was not segmented, it was monopartite, it was a single genome. This one is segmented, and we'll see that segmentation and its effects. Influenza types, as mentioned before, now with just a little more detail. Type A is known to be the most severe form. It causes pandemics and epidemics. It also infects other animals. And for that reason, if you have broader host range, it means you can change a lot. And you'll see this with the rotaviruses as well. I don't know if you've covered that. So the broader the host range, the number, or even the more the number of infections a virus does, then the more likely it will change or mutate or become different. And the next host infected may be more susceptible because the virus has changed. The second type, which is type B, causes outbreaks, and these are usually seasonal epidemics. It may also infect seals, but the zoonotic form of type B is not so strong. And it tends to cause outbreaks in institutionalized communities, and especially in children. So type B is important. Sometimes it causes as many as half of the number of cases of influenza in a year or in a particular outbreak. But type A is one that we worry about the most because it varies a lot and it's likely to cause more severe disease. Type C, we don't follow it much because it doesn't cause a severe disease, and type B causes disease in cattle. So for influenza A, there is a further classification, which you may have seen before, like you may have heard of H1N1 or H3N2 or H1N5. What are those based on? These subtypes of influenza A, they're only for influenza A, not for B or C or D. They are based on the surface glycoproteins that we'll talk about briefly. And these surface glycoproteins are both H and N, H for hemagglutinin and N for neuraminidase. And for hemagglutinin, you can have the hemagglutinin having 18 subtypes. So they are called H1, 2, 3, all the way to H18. Neuraminidase can have 11 subtypes, H1 to N11, N1 to N11. So what this means is that somebody might be infected with H1N1, H1N2, H3, and H1N3, all the way to 11. And the permutations you can see are in the hundreds. Actually, I think more than 130 something permutations have been found in humans. Not all these have been detected in humans, some have been detected in animals. And usually in a season, only 1 or 2. You can see currently, whatever is circulating is H1N1 and H3N2. You may remember from Makuru story that whatever was circulating was called H1N1 PDM09, which is really H1N1 that changed in 2009 to cause a pandemic, hence the name PDM09. So the combinations are many, but usually whatever is circulating at a greater level is usually 1 or 2 or 3 viruses. So this type of classification is highly functional because it's what we use to identify the viruses, and you'll see later. Even here you can see examples, like for example, Air California 7, 2009 H1N1 virus. This has a meaning, but it's really how to call, this is the full name of a virus, an influenza virus. And you can see here, type A, influenza viruses can have any one of these combinations of hemagglutinin and neuraminidase, and those would be subtypes. And type B has only 2 lineages, and the 2 lineages, now they are not usually, they don't go by the name subtypes, they go by the name lineages, Yamagata and Victoria, and they cause seasonal epidemics, especially in children. You can see an example here, B Washington 02/2019, B Victoria lineage virus. And then you can see B Fuke in a type of the Yamagata lineage virus. I will explain to you briefly what these numbers mean, so don't worry about them. The idea here is you have type A being, you have influenza A being classified into different subtypes depending on the surface proteins. The surface proteins are hemagglutinin and neuraminidase. Again, I'll show you how they look like briefly. Type B or influenza B is classified into 2 lineages, Yamagata and Victoria. So these are like subspecies level categories. You have the genus, you have the species, and now we are for type A, we have subtypes. For type B, we have lineages. And you can even go further to clades, but we'll stop at lineages. This is what the name means. So when you see A Sydney or Nairobi 0597, what does this mean? A is the type of the virus. So it will be A, the place or location where it was first isolated, the number of the strain. So maybe in Sydney, they had 10 strains that year. This was the strain number 5. The year which it was isolated, this is 97. It could be 2012 or 2020. And then the subtype. Is it H1N1, H3N2 in this case? And this is the full name of the virus. Usually this is important because we use it for identifying which vaccine we should give. But for use, generally you hear people talking about H3N2 virus. You may find another H3N2 virus isolated in Nairobi in 2020. It will be different because of antigenic variation. So not all H3N2 viruses are the same. A good example is the one I've just told you now, H1N1 is still circulating, but does not cause a severe diseases, H1N1 PDM09, because PDM09 changed from what was existing, what had been circulating. So keep that in mind, that even within a subtype, we will have different strains. Now, the influenza virus structure, feel free to post any questions if you feel there's something you do not understand. The influenza virus structure looks, as you can see here, from the inside you have these segments, which are 8, of RNA, which is coated with nucleocapsid, and so it is called nucleoprotein because it contains both the genome and the protein covering it. And you can see 8 segments, each coding for different proteins. And with this is contained within the core, and the core is surrounded by this brown matrix protein. And the matrix protein has 2 proteins. The brown one is called M1, and the M2 protein, which is purple, is important because it's an ion channel which assists the virus when it comes to uncoating, and we also target it for treatment. Although the drugs we use for this are now not very effective due to resistance, but this purple channel, ion channel, is called M2 ion channel, and it is important when the virus is uncoating. I'll show you a short video how this happens. The next layer is the light blue layer, which is the viral envelope, and it's derived from the host as the virus is budding out. And you can see it is studded with 2 types of proteins. The yellow is hemagglutinin, and the green is neuraminidase. These are the surface glycoproteins. Remember, we just described that hemagglutinin has different subtypes as well as neuraminidase. And maybe the next slide will explain actually more what these structures are for. You can see, as I said, the different segments of the genome, and this is important for the virus, and it helps even with antigenic variation. So for the envelope, the 2 important structures here, hemagglutinin is used for attachment onto sialic acid, which is found, or some people also call it neuraminic acid. Sialic acid is found in plenty on the epithelium, or the cells lining the respiratory tract. So sialic acid is expressed a lot in the mucus of the cells, and neuraminidase is an interesting enzyme produced by the virus to clear sialic acid. So how come the virus is clearing its own ligand? Why is it interested in clearing the ligand that it binds to? Remember, this sialic acid is in plenty, or neuraminic acid is in plenty. You can call this sialidase if you like. The virus, once it attaches to that sialic acid, if the sialic acid is attached to a cell, the virus will enter that cell. But if that sialic acid is in the mucus, in the lining of, or in the epithelium of the respiratory tract, what will happen is that virus will no longer be available to attack a cell. It will be bound in the mucus and coughed out. So in a way, when a cell has been infected, when the viruses are being released, it is better for the virus if there is no sialic acid around that cell, because then it will just bind those sialic acid molecules and not attach to other cells. So it produces neuraminic acid, neuraminidase to break down sialic acid, so that this can facilitate viral release. So if this is a cell, and the cell that is infected is now releasing these viruses, the new viruses can be free to go and bind other nearby cells without being trapped by sialic acid. So it's an interesting relationship. You also have, and I hope that's clear, you also have, and in fact, the most effective drugs today for influenza target neuraminidase. So if you block neuraminidase, then you block virus release. The matrix proteins, as I said, are the brown and the purple. We are most interested in the purple one, which is the ion channel, which facilitates uncoating the virus. And if you block it, then you block uncoating of the virus. And we have drugs that block uncoating. The RNA segments you can see here code for different proteins, and the fact that it's segmented allows for something we call reassortment. We'll talk about that in a few slides when we're talking about antigenic variation, but it's very few viruses actually have segmented genomes. A good example is rotavirus. There are few bunyaviruses like Rift Valley fever, and such kind of viruses tend to undergo very sporadic genetic reassortment that can generate very different viruses from what is existing. There are few non-segmented viruses that can still undergo reassortment, but that is more rare. It's not as common as for these other segmented viruses. You can see if you have to take a photograph of influenza in using an electron microscope, you can see here the shapes. I don't know how you describe this shape. Anybody? The one is zoomed in, one is zoomed out a bit, but all these are virions within a sample or a culture, actually, that are being visualized using an electron microscope. How do you describe this shape? Bean shape. Thanks, Kamunya, for responding to Laura. P-shaped, bean-shaped, cashew nut. You can see they are different shapes. So for that reason, we say this shape is irregular or pleomorphic. So when you see a virus that looks like irregularly shaped or pleomorphic as different shapes, then we say that could be influenza. Now, replication. I will explain this, then we'll look at a video so that we understand it well. The first step of replication, of course, is attachment. And you can see the influenza virus or virion here attaching to sialic acid on the epithelial cell. Is it blue or gray? I don't know. This is an epithelial cell. And once it attaches, it causes an endosome to form. So it enters the cell through endocytosis. After entering the cell through endocytosis, the endosome is acidified. And the acidification you can see is due to these lysosomes. I don't want to focus too much on that process, but the acidification facilitates uncoating because the hydrogen ions go through the M2 ion channels. Hydrogen ions are in the acid. So with that acidification, you have uncoating. But you can block this process of uncoating, meaning that the genome will not be available to go into the nucleus. And this brings me to an important point. RNA viruses, as we've said before, will generally replicate in the cytoplasm, except for two families. The first family is Orthomyxoviridae, which is influenza viruses. And the second family is, anybody? HIV, exactly. That is Retroviridae. So HIV and influenza are RNA viruses that replicate in the nucleus. Other RNA viruses will replicate in the cytoplasm because there's plenty of RNA polymerase in the cytoplasm. DNA polymerases or DNA viruses will go into the nucleus because that's where you find DNA polymerase. Okay, except for Poxviridae, which carries its own polymerase, so it can replicate in the cytoplasm. So after uncoating, the genome enters the nucleus, where you will have both. Initially, of course, you have the virus, which is negative sense RNA, being copied to positive sense. Then those are used as copies or templates to make more negative sense and positive sense genomes. And you can see here you're starting with viral RNA. You make complementary RNA, and these RNAs that are being made will go into the cytoplasm for translation into protein. But some of the proteins are required in the nucleus to make more copies of the genome. These include the polymerase and other import-export proteins, and also the nucleocapsid to coat the genome. So you can see the initial replication, the initial copies go into the cytoplasm, they produce proteins. Some of the proteins will come back to the nucleus to continue with the process of making more copies of the genome, and these copies will either become templates for protein synthesis, or they will become the genome for the next copy of virus, and they are coated with the nucleoprotein or the capsid, which then will be exported into the cytoplasm and assembled together with the other proteins now to make new copies. So you can see the procedures here, and I hope you understand them, from attachment to endocytosis to uncoating to replication to transcription. And I think I should bring transcription before replication, and translation, assembly, and budding. Transcription is basically making copies of the genome to messenger RNA so that it can be translated into protein. We'll watch a short video on this. Let me just change this. We'll watch a short video on this so that we can understand that process better. I need to share my sound. Just a minute. Okay.
The flu is a contagious respiratory infection caused by influenza viruses. Like all viruses, influenza must gain entry into host cells to replicate. The process involves several stages, from attachment and entry to viral RNA and protein production to assembly and release from the host cell. An influenza virus attaches to a cell when hemagglutinin, one type of protein in its viral envelope, binds to a host receptor. Hemagglutinin recognizes receptor polysaccharides that terminate with sialic acid. The cell then takes up the virus by endocytosis. The endocytic vesicle then fuses with a lysosome. A lysosome contains digestive enzymes and an acidic interior, and typically digests contents taken in from outside the cell. Unfortunately for the host, the virus uses the acidity to its advantage. An ion channel allows protons to enter the virus. The acidity inside the viral particle disrupts protein-protein interactions, causing matrix proteins to detach from proteins covering the viral RNA genome. This detachment is the beginning of the uncoating process. It's interesting to note that one type of anti-influenza drug blocks this ion channel. The acidity also triggers hemagglutinin to make a structural change and insert itself into the vesicle membrane. In this new form, hemagglutinin stimulates membrane fusion. The RNA genome and associated proteins can flow freely away from the matrix proteins into the cytoplasm, completing the uncoating process. The 8 RNA segments of the influenza genome are covered with nucleocapsid proteins, abbreviated NP, and attached to several other proteins that make up the viral RNA polymerase. These RNA protein units enter the nucleus via the nuclear pores. The RNA segments are minus-strand RNAs, meaning that they're non-coding strands. Each of the 8 segments needs to be copied into a complementary plus strand by the viral RNA polymerase before it can be used for translating proteins. To make a messenger RNA for translation by host ribosomes, the virus needs to add a structure called a 5' cap at the beginning of the mRNA transcript. The virus performs a stealthy act known as cap snatching, in which the viral RNA polymerase cuts off the cap from one of the host cell's own mRNA molecules and uses the cap to start transcription of viral RNA. The new plus-strand viral RNA gets a poly-A tail that aids in mRNA stability. The new viral mRNA leaves the nucleus for the cytoplasm, where the host cell's ribosomes translate the mRNA. In this example, the mRNA codes for the nucleocapsid protein that will coat future viral RNA molecules. Several other mRNAs code for viral envelope proteins. Envelope proteins are made by ribosomes on the host cell's endoplasmic reticulum. This particular mRNA codes for hemagglutinin. Back in the nucleus, the newly made nucleocapsid proteins prime and stabilize viral RNAs as they're being synthesized into full-length plus strands, this time lacking a cap and a poly-A tail. Newly made RNA polymerase proteins enter the nucleus and participate in the production of additional full-length minus strands that will serve as the RNA genome segments of new viruses. From these events, 10 different types of proteins are synthesized from 8 RNA segments, and new minus-strand RNA genome segments are produced. The RNA genome segments, complex with RNA polymerase, nucleocapsid proteins, matrix protein, and packaging proteins, are exported from the nucleus. The envelope proteins made on the endoplasmic reticulum move by transport vesicles from the endoplasmic reticulum to the Golgi apparatus and finally to the plasma membrane. The RNA protein complexes are then packaged into progeny viruses as they bud from the cell membrane.
Okay. So that explains the replication cycle. That video will be in this PowerPoint. If you want to look at it again, you may want to see it a few times to fully follow the process. And now we move into antigenic variation. There is a bit on the chart. Sialic acid is essential for binding of the virus. Okay, yeah, somebody has explained there. Thanks for that. Okay, I think a lot of the issues have been handled. I should be sharing my salary with some of these guys. They're doing my job very well. The underlying principle of antigenic variation is immune evasion. And this happens because the viruses, by random chance and natural selection, will, as they mutate, if the mutation gives them the benefit of varying or antigenic variation, they tend to be selected because other viruses that do not vary their antigens or the surface proteins will get killed or destroyed by or neutralized by the human antibodies or by the host antibodies. It could be a chicken or a pig. So those viruses that manage through chance or through mutations which occur by chance to vary their proteins, then it means they'll not be able to, they will not be amenable, they will not be sensitive to the existing antibodies. And for that reason, they'll be able to multiply and infect new hosts. How does this happen? There are two types of antigenic variation mechanisms. The first one is an antigenic drift, which occurs gradually, steadily, and causes actually most seasonal outbreaks. And it will occur in all influenza viruses due to accumulation of mutations. So every time the virus infects a new host, every time the virus multiplies, after a certain number of insertions, every time the RNA polymerase inserts a base, it could be maybe after 1 million bases, it makes an error, inserts the wrong base. Remember, the RNA polymerase does not proofread as well as like the DNA polymerase does. The DNA polymerase is very good proofreading capacity. RNA polymerases don't have that. So for that reason, whatever errors the polymerase makes are carried forward to the next generation. Those errors could mean that the next generation will die off, or there will be no effect, or they will have an advantage. The advantage could be drug resistance, it could be escape of existing antibodies. As we saw with COVID-19, new variants were able to escape existing antibodies and cause infections, even in people who have been vaccinated. So this gradual accumulation of mutations, you can imagine after several copies are made, there are mutations. As the virus infects the next person, there are more mutations. And eventually, if I started, say, a strain, I infected somebody else who infected the next person, by the time this virus comes back to me, it's slightly different from what I had before. So the antibodies that I have may not neutralize the new strain that has been generated by this accumulation of mutations. And for that reason, the immunity that I had either from previous infection or even from vaccines becomes ineffective, and I may get infected. Maybe I will not have very severe disease, depending on how much the virus has changed, or if I have other underlying issues, I might actually get severely sick. And this is what causes seasonal outbreaks and may cause epidemics. The next, this is an image of variation, just showing you that the gene has changed and the shape of the protein, the surface protein has changed from this triangle to this square or rectangle, just to show that if an antibody was binding this one, it will not bind this one very well. This is just for illustration purposes. So really, antigenic drifts occur due to accumulation of mutations. And this occurs, it's normal for RNA polymerases to make mistakes. Many of the mistakes actually make the virus unfit, so the virus might die off, but some of the mistakes give it an advantage, and then the power of natural selection or the laws of natural selection take effect. Isn't it their nature to mutate? Yes, it is. Viruses will mutate a lot because of the errors they make, and the more copies they make, the more chance for an error. So the more people are infected, the more chance for mutation. Okay. Antigenic variation can also occur due to the second mechanism called antigenic shift, and this is really due to genetic reassortment. Remember the genome segments, the 8 segments of influenza A? Because of those reassortments, if one virus, we call it, if one host cell, we call it a mixing vessel, is infected by 2 different viruses. So for argument's sake, let's say you have H1N1 and H5N5 infecting one cell. These viruses will continue to make copies of their genome, copies of their protein, and as they assemble or as they get assembled, there could be mixing of the different genomes. So you end up with a new virus that has picked some segments from H1N1 and some segments from H5N5. So it might end up becoming H5N1 or H1N5, for example. It could still be H1N1, but has some genetic components from H5N5 or vice versa. So really, this is where the genetic segments are mixed within the new virus so that you have a hybrid form of the virus that is new and that no one has been exposed to before, which means there is no existing immunity, and for that reason, the virus can easily cause a pandemic across the world or very large epidemics. Fortunately, this is not very common. It occurs sporadically. At some point, we thought it occurs every 100 years, but now we know it occurs more frequently. A lot of times, the viruses may not be noticed, may not cause a large enough outbreak to be noticed, but sometimes they cause a serious problem. And this is because they form a totally new subtype, and there's no existing immunity. So not just the elderly and the young are at risk, even the elderly and the very young are at risk, even the healthy adults like you guys are also at risk. I hope that makes sense. So to differentiate, antigenic drift is due to gradual accumulation of mutations. Antigenic shift is sporadic, it is sudden, and it occurs due to the assortment of RNA segments. This here is a cell infected with 2 different viruses. During assembly, the genome, the new hybrid virus picks genomes from both infecting viruses to form a new subtype of the virus. It could be a pig, actually, infected with pig and human influenza, and then that forms a new influenza that is able to infect humans, but also has some proteins from the pig virus. It could be a chicken, same thing. It could also be a human, but generally we find it in pigs and chicken because we find farms that have hundreds of thousands of these animals together. And remember, the more the hosts and when they are close together, the more likely that the virus will have a chance to mutate because it's infecting more hosts. That's why, for example, when the viruses get into cities, they really can cause a lot of havoc. And you saw with COVID-19, a lot of the cases were in cities where there are more people, so it's easier to spread, and there are many more opportunities to mutate because each person it infects is an opportunity to change its antigens or surface proteins. The other thing about viruses is pathogenicity. Some of the viruses you hear are being called highly pathogenic because they cause more severe disease. Others will be called low pathogenicity. And the other thing is infectiousness. Some will be highly infectious, others will be not as infectious. So if you have a pathogen that is highly infectious and has high pathogenicity, then you are in trouble. What we see often is that avian flu or a virus that is coming from chicken or birds is not able to infect humans very well or very efficiently. But the few that it infects, it kills, like half of them. So that is highly pathogenic, but of low infectiousness. If it mutates, if it keeps infecting humans, it will perfect its ability to infect humans. And once that happens, it might retain its pathogenicity or it might lose it, because every change has a cost, every mutation has a cost. But if it retains the high pathogenicity and gains high infectiousness, then that would be a very worrisome subtype or strain. Now for pathogenesis, transmission is usually from small particle aerosols. If you're in a room, say a lecture hall, these aerosols are very small. They can even travel to the back of the class if it's dry enough and it's not so humid, because they are very tiny, 40 meters away, and they'll remain in the air for long times, as compared to droplets. A droplet will remain in the air for a few minutes and then drop because it's heavy. But aerosol will remain in the air for long. And the incubation after infection or exposure is about 2 to 3 days, 18 to 74 hours. It will replicate in the epithelium of the respiratory tract, usually in the upper respiratory tract, in the pharynx or in the throat, sometimes in nasopharynx as well. And rarely will it cause viremia. So what it does is that it spreads contiguously. So influenza rarely causes viremia. You check, this is less than 1% of, maybe babies might get it, but most people will not have viremia. So if you're doing a PCR, for example, or you want to culture the virus and you take blood, you won't find it. But that doesn't mean the person is not infected. We'll see which samples or specimens are good. So as it replicates in the epithelium, it spreads contiguously, basically to the nearby cells, continuously until it goes from the upper respiratory tract to the trachea, bronchi, all the way to the lower respiratory tract, and can cause disease or even issues like viral pneumonia in the lung parenchyma, in the lung tissue. The cell death from influenza is due to direct viral effects and also from cytotoxic T cells. What this means is that the cells that are infected with influenza will be destroyed, but they'll be destroyed by the virus, but also the body's immune system is triggered and it will activate cytotoxic T cells. I don't know if you've covered immune response to viral infections, but what happens is generally when killer T cells are activated, they will release enzymes that make holes on the host cell that is infected and induce apoptosis. So basically killing the cell that is infected. And so if you're killing lung parenchyma cells, the pneumocytes, what happens is that you're going to interfere with gaseous exchange, you're going to cause a lot of inflammation, fluid leakage and cell leakage, debris. So basically the lung becomes consolidated, becomes solid, rather than allowing gaseous exchange. It is now inflamed, it will cause painful breathing, it will not allow very good gaseous exchange, And so the person will have all these symptoms of pneumonia. The cells that are lining the epithelium, because they're also destroyed, that interferes with clearance of pathogens. If remember, these cells are lined with cilia and mucus producing cells, so they trap virus and other pathogens like bacteria, like the throat, which has a lot of the normal flora bacteria. Some of them would love to go down, but this mechanism protects you from those bugs in the throat because it keeps clearing them. And so if that mechanism is interfered with, it's like if you leave your door open, and if you're living in the hostel, you know how risky that is. So you've left basically removed your first line of defense. And what happens is that you open up your body to, or your lungs to, infection with other pathogens, especially bacterial pathogens that are in the vicinity. Usually, the initial viral clearance is by interferons. As the lung cells become infected, or as other epithelial cells become infected, they will produce interferons which warn nearby cells, and so the virus is not able to spread as much. And usually this is enough to clear the virus through cell-mediated immunity. But sometimes when this fails, then the patient goes into more symptoms, and then they can get very sick. Usually there's no antibody response early in the infection, but the person will produce antibodies, and if they're exposed to this same virus in the future, they will be protected. But remember, we just talked about antigenic variation. So if the virus changes its surface proteins, it will be able to infect this same person regardless of their previous infection. Although there may be some partial protection, meaning that the disease may not be so severe unless the virus has changed so much. And we are seeing a lot of that even with COVID now, where people who were exposed before become sick, or they become infected, but they don't get very sick. I'm trying to look for a list of bacterial pathogens that I needed to show you, but it's okay, they'll come later. And what I need to underline on these slides on pathogenesis is this. The virus gets into the body, it will destroy cells in the tract, in the respiratory tract, and also it will trigger the immune system to cause further damage as the immune system tries to clear the virus. If the immune system succeeds to clear the virus, you're okay. But if the immune system does not succeed, then you may get into viral pneumonia or other forms of pneumonia like bacteria, which may actually take advantage of the destruction of the epithelium to cause infection. So let's see this question, which you can answer in the chat again. One of the following are key in the pathogenesis of influenza virus except. So which one of these is not important in pathogenesis of influenza virus? Viral cytopathic effect, interferons, cytotoxic T cells, and viremia. I can see most people are getting the answer correct. So it means you guys are still alert. That is good. We can proceed. Yes, there's no role. The viremia plays a very minor role. As I said, less than 1%, or very few people will have viremia. Most people will not. So clinical features, I would like to highlight here that these clinical features are not specific enough for you to say this is influenza. You can only suspect. For example, when there's a spike in COVID, you can see symptoms and say this is most likely COVID, but the only way to be sure is to test. And that's the same thing with influenza. And we are more worried about influenza in the high-risk groups, especially the very young, usually under 1 year, and the elderly, and also the sickly. And the sickly are usually people with chronic diseases like renal, heart, and lung diseases. Because they tend to decompensate fast and can die. A lot of deaths from influenza occur in the elderly. And the main causes of death is bacterial pneumonia, remember, due to the loss of the protective mechanisms, and also heart failure in people with congestive cardiac failure or other heart diseases. They may end up dying because now the heart is being overworked because the lung is not oxygenating properly, and this becomes an issue. And complicated symptoms will include things like headache, fever, muscle pains, cough, rhinorrhea. And interestingly, from 2009 with the PDM 09H1N1, we saw GIT symptoms in some patients, which we think are due to cytokines. Remember, cytokines are the things that you produce when you're sick. They're the ones that make you have a lot of pain, have a fever, have nausea, have vomiting, and even diarrhea. So that is a chemical response to, functional response to the cytokines as chemicals or as produced by the body. There could also be pulmonary complications, and this could be, especially in the very young, swelling and even obstruction of the upper airway, which causes croup, where the larynx, the trachea, and the bronchi are inflamed, and for that reason, they could cause obstruction. It's called laryngotracheal bronchitis, and these babies or children will breathe out with a sharp whistling sound because of that obstruction. The viral pneumonia occurs when the lung parenchyma is attacked, when the lung cells, remember the lung is made up of pneumocytes. And so when they are attacked, they get destroyed. Then you get viral pneumonia. But you can also get bacterial pneumonia because of the loss of the protective effect of your epithelium. And usually the common causes of bacterial infection are Haemophilus influenza. Influenza as a disease was identified, as I said, maybe in 1500 or something, but we didn't know what was causing it. Later, some people were isolating pathogens. When we learned after Koch's postulates, people were isolating pathogens from all diseases, and a lot of people were turning up. The people with influenza were turning up with Haemophilus influenza, this bacteria. And although it is not, initially it was thought to be the cause of influenza because it was in so many people, but of course we know it was a super infection. It was an additional infection on top of the virus, but at the time we could not see the virus. So we said this is the cause, or somebody did. But remember, it's not the cause of influenza. It actually just complicates the infection. Streptococcus pneumonia is usually found in the throat and can cause pneumonia. Fortunately, now we have vaccines for it. So because it's a severe disease, but it's also very easy to treat with amoxicillin and other penicillins. Staph aureus, a lot of it also in the throat and even in the mouth. So it can go down and cause lung disease when the mechanisms of protection have been lost. This is the story of pneumoflora becoming infections. And remember, a lot of our infections are usually endogenous. They come from pathogens within us that are normally pneumoflora, but when there's an opportunity, they become pathogenic. You can also have myositis and glomerulia in a few patients. There are non-pulmonary complications that you should look at. I won't take too much time with this, but myositis, cardiac issues, encephalopathy, Guillain-Barré syndrome, and Rice syndrome. This is in the liver, and GBS is ascending paralysis due to destruction of the myelin on the nerves, and you of course know what encephalopathy is. Cardiac complications usually due to compensation. Then muscles could also get inflamed as well. How do we diagnose influenza? I will take a short break here as I look at the chart. Sir, can being reinfected by a completely different strain of influenza cause any syndrome like hemorrhagic shock syndrome? Well, we haven't seen, I don't know what you mean by hemorrhagic shock syndrome, but we haven't seen a lot of sepsis like, or where people get shock. Shock is basically when your cardiac circulatory system is collapsing because you have insufficient fluid or you have a lot of dropping blood pressure due to, say, cytokine or cytokine storm or whatever it is that is causing you to lose blood pressure, and so you don't get enough supply into your body. And it can be caused by many things, but this is not one of the things we see a lot in influenza. Does somebody with influenza cough? Yes. And anybody with any infection in the upper respiratory tract, they will cough. Because of that, they're trying to expel that pathogen, there's inflammation and all that. What did he say was the reason for the two RNA viruses to replicate in the nucleus? The reason is, I didn't say it, but the reason for influenza, I think, is because it requires to process its RNA in the nucleus that it would not be able to do so in the cytoplasm. And usually, I don't know if you remember this, but you saw something we call cap snatching, where it was basically attaching a methyl cap on the 5' end, on the 5' side of the messenger RNA. That process is important and doesn't occur in the in the cytoplasm. So it means there's virus in the cytoplasm. There is a good chance that it's being copied, but it is not functional. The ones that are functional are the ones that are in the nucleus. What is the difference between rhinoviruses and influenza virus? Rhinoviruses are enteroviruses. Influenza viruses are orthomyxo. Enteroviruses include things like hepatitis A and polio. So, and the good thing is that rhinoviruses are mostly mild. They don't cause very severe disease in most people, but they do cause infections. And unfortunately, a lot of people with rhinoviruses end up being treated with antibiotics and things like that, which causes or worsens our problem of antibiotic resistance. The non-pulmonary complications due to encephalitis is also due to cytokines. Yes, there's that, but also there are other immune antibody complex deposition that cause issues in the body. And that's why I want you to read on that if you're interested. It's a bit deeper, and I don't want to waste too much time going into it. The importance of clearing sialic acid is to clear the way. It's basically, you can imagine if you are a bouncer. The role of this bouncer is to let you into the club, but prevent you from getting out, so that you can maybe drink as much as possible. So when you're trying to get out, you can hire people to get rid of the bouncers so they don't catch you and take you back in. So or make you not leave. So sialic acid clearance will make sure that the new viruses that don't need to attach to that cell that is producing them will find their way out of that cell and attach to other nearby cells. If there's a lot of sialic acid hanging around and we see a lot of it in mucus, then the virus will just be trapped into the mucus and they'll not be available to attach to other cells. So that is the point. I will look at the last comment, then I can go into diagnosis. Dry cough, influenza, normal cough, or rhinovirus. It's very difficult to differentiate the cough, so I won't go into that. The diagnosis, except for duration. If somebody has been coughing for too long, then you know either they have a chronic disease or they have TB. If it's a few days, it can be anything. Diagnosis is mainly clinical, actually, a lot of times. And you suspect based on the epidemiology, but for you to confirm, you need to get swab specimen, and the specimen is usually throat or nasal swabs or both, and so you get a swab maybe for the nasopharynx. I think I'll show a video to collect that so that you do it correctly, and then another one for the throat. Please don't use the same one from the nose to the throat. And then some people also do nasal washes. So maybe I should try and show you the video now, then we can proceed with the presentation. Just making sure I don't lose my, yes, we are here. You should be able to. So just confirming that I'm sharing. Yes, I think I am.
It is recommended to wear appropriate personal protective equipment, gloves, surgical mask, eye protection, when collecting samples. Wash hands. Put on gloves and surgical mask. Open the package containing the sterile PureFlock Ultra Swab and aseptically remove from package. Tilt the patient's head backwards slightly to straighten the nasal passage from the front of the nose to the nasopharynx to make insertion of the swab easier. Insert the PureFlock Ultra Swab into one nostril straight back, not upwards, and horizontally to the nasopharynx up to the measured distance on the swab handle. The distance from the corner of the patient's
Just to note, please make sure your head is tilted back. If you ask me, this position is a bit too forward, so I would probably sit them up and have the position tilted quite back so that you go straight to the sort of the back of the ear.
Nose to the front of the ear gives an estimate of the distance the swab should be inserted. Rotate the swab up to 5 times and hold the swab in place for 5 to 10 seconds to collect sample material. Insert the swab into the viral transport medium. Break the swab handle at scored breakpoint line.
Also note that swab is very tiny, not as big as, I don't know if I have one here. So there are some that are quite thick that will not fit in the nose. So be careful. You might injure somebody if you insist on putting say a high vaginal swab into the nose, or even the one used for the throat swab is a bit thick. This is very narrow to make sure you don't cause injury on the mucosa of the nasal mucosa.
Replace cap and tighten to secure. Remove and dispose of gloves and mask properly. Label vial with appropriate patient information. Deliver samples to laboratory for testing. It is recommended to wear appropriate personal protective equipment, gloves, surgical mask, eye protection, when collecting samples. Wash hands. Put on gloves and surgical mask. Have the patient facing a strong light to ensure the area to be swabbed is visible. Open the package containing the hydroflox swab and aseptically remove from package. Have the patient tilt their head backwards, open their mouth and stick out their tongue. Use the sterile hydroflox swab to swab both tonsillar arches and the posterior nasopharynx without touching the sides of the mouth.
Remember, if you touch the side of the mouth, you'll be collecting other pathogens. You're not just interested at the back of the throat.
Break the swab handle at scored breakpoint line. Replace cap and tighten to secure. Remove and dispose of gloves and mask properly. Label vial with appropriate patient information. Deliver samples to laboratory for testing.
So I hope that helps. If you get a chance to collect that specimen, you can always review those videos again. Now, once you get that sample or that specimen, you have the option of testing this, and this could be at the clinic actually or the bedside, antigen detection, which is rapid. And it's what we mentioned before, if you remember, low sensitivity, high specificity. What does this mean? Low sensitivity means if it is negative, it does not mean that person has no influenza. It could be a false negative. Why? Because maybe there is not enough antigen. If it is positive, this tells you that that person is most likely having influenza. It will not give you a lot of false positives. When it says the person is positive, then most likely that person is positive. So it means if you have the means, you should repeat tests that are negative, but the ones that are positive, you don't need to repeat. You can treat that person confidently because you know it is not likely to say somebody is negative as positive. But if it says this person is negative, there's a good chance, almost 50%, that the person is actually positive. So you need to understand these concepts of sensitivity and specificity to learn how to use them to your advantage. The other good test, which is a very good sensitivity and specificity, is PCR. And PCR has actually, over the last few years, now become highly multiplexed so that you can test 12, 16, 20 pathogens at once. if we go back to our first slide or second slide, we talked about many pathogens causing influenza-like illness. So if you just test influenza or PCR and it's negative, do you send the patient back to test for COVID? You send them back to test for rhinovirus. If you're testing all these viruses together and even some bacterial pathogens in one test, although right now they're very expensive, I think they'll become cheaper, then you're able to make that decision at once. So multiplex PCR tests are encouraged for this kind of thing. So the same sample can go for a PCR test within a day. You're able to tell if the person has which respiratory tract pathogen the person has. There are also serologic tests. These are mainly immunofluorescence or ELISA, but you can also do hemagglutination inhibition assay. And this is based on, I think I'll show you the next slide just to describe this more, but it's based on the hemagglutinin protein. The hemagglutinin protein is so named because it causes agglutination of red blood cells in vitro, not in the body. But if you put them virus on a petri dish together with red blood cells, it will cause hemagglutination. And so this is, and it's not the only virus that causes that, by the way, there are others. So this is one of the tests we can do, and I'll show you how. Virus neutralization is basically taking the patient's blood to see if it has antibodies against viruses. And you usually do that by putting live virus together with a blood sample and then culturing it over time. This needs to happen in a basic biosafety level 3 at least. You can also do viral cultures, which any microbiologist will tell you cultures are probably the best way to identify a pathogen. So of course they take time, they're costly, they require different security levels. And for influenza, this will require at least BSL-3. So it's not frequently done. So hemagglutination inhibition assay works as you can see here. Imagine these are 3 different wells. In the first well, you just put RBCs and nothing else. So there's no reaction, there's no hemagglutination. In the second well, you put the virus, influenza virus, and RBCs or red blood cells, and so the virus will cause hemagglutination. In the third well, so these 2 are controls. In the third well, you add the patient's blood. So this is a virus that you know this is a virus you have stock of. You know when you add it to RBCs, it will cause agglutination. And because you know the virus, when you add the patient sample, if the patient sample has antibodies, they will prevent this virus from binding the red blood cells, and so there will be no agglutination. So you will know that the antibody or the patient's plasma or serum has prevented agglutination, meaning that that patient has antibodies against this virus. So if this was H3N2 or H1N1, and the patient's blood is able to neutralize it, then you know that patient has that specific virus. I'll take that again. You are mixing a known virus, you have it, it's like a control, with RBCs or red blood cells. And what you expect is agglutination. But if you add to this mix the patient's blood, if the patient's blood has no antibodies, agglutination will still occur. But if the patient's blood has antibodies against that virus, agglutination will be inhibited. So that is how hemagglutination inhibition assay works. You can see the clinical bit, you cannot forget it. This is, I know you guys have not started looking at x-rays, but this is a normal chest x-ray with very nice lung fields. With pneumonia, if you get a viral pneumonia like influenza, you may start to see these solidifications or opacities because of inflammation. And this means that the person is not oxygenating well, they may be in pain, and so on and so forth. So keep in mind that we are teaching you laboratory diagnostics, but you need to have your clinical. Over time, of course, you get to learn your clinical aspects as well, which you combine with your lab diagnostics to make a determination as to what the patient needs. Treatment, I can see quite a bit on the chart. A lot of questions on shift and drift. It's a good discussion. I think I'll clarify that later. Kuna mtu ana maoni. What occurs more frequently is the drift, because mutations occur every day. The shift occurs sporadically once in a while because it requires genetic reassortment. So how do you treat influenza? The first thing is supportive care. If this person has a fever, if they have pain, treat that. If they require oxygenation, give them oxygen. If they require mechanical ventilation, then they require ICU to get supported breathing. And sometimes they may have other complications that require even ionotropic or cardiac support. Chemotherapy is 4 drug categories. The most commonly used or the most probably effective right now is viral release inhibitors. Remember, these are the guys who inhibit neuraminidase. And acetaminophen should be the first. We know it as Tamiflu because it is given orally, and it is the most widely used now of these 3. And the polymerase, the viral release inhibitors work for all the influenza viruses, A and B especially, because that's our focus. But you also have polymerase inhibitors. And these polymerase inhibitors used for influenza virus are actually broad spectrum polymerase inhibitors. You might see them in hepatitis C. Some people have tried them for other viruses, but they're effective and have been licensed for use with influenza viruses. Ribavirin and favipiravir. Favipiravir is quite, well, not so recent, but more recent than ribavirin. And you can also use uncoating inhibitors. Unfortunately, uncoating inhibitors are, there's a lot of resistance against them, and they only work for influenza type A. They don't work for influenza type B, and even for type A, there's a lot of resistance. So they have fallen out of favor really due to drug resistance. Recently, maybe a couple of years ago, a new drug was licensed. It's called baloxavir. And baloxavir is an endonuclease inhibitor. Now, endo means inside. Nuclease is an enzyme that cuts the nucleic acids. The role of baloxavir is to stop the cap snatching by influenza virus. If you remember, the influenza virus polymerase has the ability to cut the cap on the host messenger RNA and attach it to its own messenger RNA. And that is called cap snatching. So messenger RNAs in our bodies usually have a methyl cap at the 5' end and what we call a poly-A tail at the 3' end. So this methyl cap assists the virus, assists the messenger RNA to be stabilized and attach the ribosome so that it can be translated. So when the virus enters the nucleus, one of the things it does is to steal our own messenger caps, mRNA caps, and attach it to its own. And the enzyme it uses is the same RNA polymerase, but as a component of it that does cap snatching because it has an endonuclease activity. If you block that endonuclease activity, then whatever messenger RNAs are being produced will not be very fit for translation to protein. So you will stop replication. And this can be done by using baloxavir. If you watch the video again, you'll see the place where cap snatching is occurring. That is the step we are referring to. You can also use antibiotics in rare conditions where you suspect or have evidence of bacterial infection. But not everybody who has, actually a lot of people with common colds will not need antibiotics. A lot of people who use the antibiotics for upper respiratory tract infections don't need them. And that's very bad because we are encouraging drug resistance. So don't use antibiotics unless you have confirmed that either through culture or through the other markers, biomarkers, maybe things you'll hear about later like CRP. You can use that to confirm if the person has bacterial infection or not. Prevention is broad, and the most important ones, I think, are vaccines. The vaccines are 2 major types. You can subcategorize this, but we'll not get into that. There is inactivated, which is basically not live, the opposite of live. You can also have live attenuated influenza virus. We don't use this as much. We are only used in children. In adults, and especially the elderly, they tend to have a lot of side effects, so we avoid them. But the inactivated vaccines are the ones that are mostly used. And they can be quadrivalent, meaning you have 4 viruses covered, or trivalent, meaning you have 3, or monovalent, meaning you have 1. Monovalent usually made or manufactured when you have a prolonged outbreak against specific viruses, and usually not for routine vaccination. But currently routine vaccination is mainly with a quadrivalent vaccine. And I'll show you an example of a quadrivalent vaccine. You can also give chemoprophylaxis. Say, for example, guys who are in an aeroplane, one of them turned positive for a new strain of influenza that is circulating. You can actually give everybody in that aeroplane chemoprophylaxis, dositamivir, within 24 hours, and that might prevent the virus from spreading. Now, every year, actually every 6 months, every February and August, WHO releases recommendations for vaccines to cover the southern and the northern hemisphere winter seasons. This child here is getting FluMist, which is live attenuated. This lady here is getting inactivated vaccine. This one usually we don't give to adults because of the side effects. So, who should be vaccinated? Pregnant women are recommended, children under 5 years and above 6 months, elderly individuals above 65 years, people with chronic conditions, and healthcare workers. Could somebody tell us why they think healthcare workers should be vaccinated? Anyone? Exposure, yes. Any other reason? You're exposed, but they may be young, so they won't die. The risk is what? They are patients. They are taking care of people with diabetes, they are taking care of neonates, they are taking care of chronically sick. So if they are sick, they will transmit to their patients. Now, you can see this recommendation for 2022-2023. It was released in February for the Northern Hemisphere. Remember, they released it in February so that by November, the vaccines have already been made and they can now be given from August onwards. And this is two types, two type A and two type B. You can see A and A. This is type A and type A, and then two type B. You don't have to cram these numbers, but you can see the type A includes an H1N1 like virus and an H3N2 like virus. Remember, these are the two viruses that are circulating, but they are different strains circulating. So you may find that the Southern Hemisphere was using something like A Sydney or A something else. So it is not necessarily the same virus, but it's similar to H3N2. The B viruses, you can see there is one Yamagata and one Victoria to cover both lineages. So if somebody is getting a vaccine for influenza now in the Northern Hemisphere, this is what they're likely to get. Now in the tropics, we have to look at our data to see what is circulating to know what is necessary. And even then it may not be available. So sometimes we end up just choosing either the Northern or the Southern Hemisphere, depending on the time. We also need to keep other things in mind, not just vaccines. Surveillance is important. And usually we look at people with influenza-like infection to see what they have, which viruses they have. Screening, for example, for people coming into the airports and other places, and even for places where you have crowding or institutionalization, screening might help. Information sharing. So if you find a new virus, you should share that with the world so that we can start responding early, educating both the patients and also the masses and also the healthcare workers who are supposed to detect and respond. Drug prophylaxis, like we saw with the oseltamivir. Vaccination should be at least annual. Culling of animals. If you have a chicken farm in Mlolongo and you see influenza, you're supposed to kill all the chicken and bury them. Limiting travel for me is usually one of those things that doesn't work very well, but if I know there's an outbreak somewhere and I don't have to go there, I can stop going there. And we know people who need to travel will still travel anyway. So it doesn't work very well, but it helps. Isolation of infected persons also helps. I need to highlight here that influenza is a zoonotic disease, and zoonotic diseases form 60% or nearly 2/3 of all human infectious diseases. And any new emerging infections, 3/4 are also zoonotic. And you can see Ebola, influenza, even HIV when it emerged. And there is also the threat of them being used for bioterrorism. And every year we might detect about 5 new diseases, and 3 of these tend to be of animal origin. That is in humans. So if we don't observe One Health, we don't keep human, animal, and environmental health taken care of, then we may not be able to take care of one part only. If you're focusing on human health without focusing on animal and environmental health, you may end up losing the battle. This here is a thermal scanner, which is used in airports to look at people when they are far. You don't have to be very close to them. If somebody is red on this scale, you can see it means they are hot, so you call them aside and find out why they are hot. And then those that don't have any issues can just go through the system. This here is the global influenza surveillance system, and these are centers across the world. There are more than 140, I think 150 thereabout, which even in Kenya we have, which people collect samples from people with influenza-like illness. They test them and find out which viruses are circulating. Then they share this information centrally with WHO, and this information is then used to inform the choice of vaccines that we need to have that year. So every year in February, this information is used to release one list for the Northern Hemisphere, and then in August, a list is released for the Southern Hemisphere to guide vaccine. And this system was very useful for COVID-19. It was used to, these labs were used to continue the surveillance or do surveillance for COVID-19. So it's a good system globally. Today, I know we've covered a lot of things. We've talked about classification. We talked about 4 genera of influenza: alpha, beta, or ABCD, alpha, beta, gamma, and delta. We talked about the characteristics, especially the hemagglutinin, the neuraminidase, the segmented genome, and the antigenic variation. We talked about pathogenesis, how the virus causes death or destruction of the cells, and also how the immune response worsens the situation in some cases. We also talked about clinical features, which are not very specific. We talked about diagnosis using rapid tests, using PCR, using serology. ELISA, although ELISA for influenza will not help you a lot unless you do at 2 points, maybe 2 weeks apart. You can see rising titers. But for antigen rapid test, you use the antigen, which will be useful. PCR is also good, especially if it's multiplex and it's being done for multiple pathogens. Treatment, we talked about 4 types of medications, from the viral release inhibitors to polymerase inhibitors to uncoating inhibitors, and finally the endonuclease inhibitors. And then for deterrence, we've talked about several things, from surveillance to vaccination to capacity building, patient education, health education. So one of the following drugs is used to treat influenza B virus. Which one of these drugs is used to treat influenza B virus? C, somebody says C. Okay, yeah, so the correct answer is oseltamivir. And one of the following is not true about antigenic shift. It occurs every year. It causes pandemics. It is due to genomic reassortment. It is sporadic, and it affects all age groups. So which one of these is false? Does it occur every year? Does it cause pandemics? Does it occur due to genetic reassortment? Is it sporadic, or does it affect all age groups? Yes, everybody is getting it right except one person. And I see here, most of you know that it does not occur every year because it's sporadic. Okay, I will look at the questions now. I see a few people with questions, so that's what I'm going to deal with now. I hope you guys will keep that kind of performance in the exams. You seem to have gotten the key concept very well. Maybe I should start up as I go down. Okay, maybe I'll just start here because I don't know how far up I need to go. Do all viruses encode their own polymerases? Generally, yes, maybe except a few, but what happens is that they use our own polymerases as well, especially for the initial replication. So when the virus goes in, they all require our own polymerase to make copies, the initial copies. Then after that, some of the proteins that the virus codes for is a polymerase. And so that, because it codes for its own polymerase, that polymerase makes the speed go fast, the replication move faster. But there are viruses like pox viruses which carry their own polymerase, so they will not necessarily require you to, will require the host polymerase. But the others require the initial transcription to come from or to be done by the host polymerases. And then after that, the viral proteins that are made include a polymerase that then means they can become, they rely on that more to make more copies of the genome. What is the meaning of segmented now? Genome can be monopartite or multipartite, meaning it could be one long segment, which is continuous, like HIV, although HIV is diploid, it has each copy is one, but it can also be, the genome can also be cut into different parts that code for different proteins, like we see with influenza. So influenza has linear genome, but the genome is in 8 segments. It's not one continuous genome. I hope that makes sense. Could you repeat specificity and sensitivity? I think you need to look at that because it's a concept that a lot of people struggle with. But if you look at how it's calculated, you'll understand. But to just repeat what I said today, if a test is highly sensitive, it means if it tells you that a test is positive, then a test is negative, then it means I don't want to cause more confusion. Sensitivity, for example, if you have 100 people who have influenza, and a test tells you that 90 of those are positive, then that is good sensitivity because it has detected 90%. But if it tells you that 50% are positive, it has low sensitivity because it has missed 50%. So if the sensitivity is good, it means you will detect most of the cases. But if the sensitivity is bad, like we saw in the influenza case that it was 60%, it means you will miss a lot of the positives. So if it tells you somebody is negative, well, positive, it's okay. But if you tell you somebody is negative, because you know the sensitivity is bad, then you will be having questions about the negative test because it could be that it was a false negative. On the other hand, if 100 people have influenza and specificity, say 100 people don't have influenza and the test tells you 10 have or 1 has, then that tells you the specificity is good because it has done, say, 90% negative out of the 100 and said 10 are positive. So it means if somebody is negative, the test is likely to tell you that person is negative. But if it tells you only half of them are negative and the others are positive, it means the specificity is bad. That if it says somebody is positive, there's a good chance that it's a false positive. So this is something you need to calculate and see how people arrive at to understand it fully. But really it's about specificity is about classifying a negative test properly or a negative person properly. Sensitivity is about detecting a positive person. So if it is the person is positive, a sensitive test will detect it most of the time. If the person is negative, a specific test will be able to say that person is negative most of the time. How long does the vaccine work for? Well, the vaccine will remain effective in your body so long as you're exposed to that same strain. But remember, if the strain changes, it will not work. So a lot of times, boosters are required every year. Annual vaccination. Okay, I don't see any other questions here. I think most of the other questions were answered. So I'll stop there. If there are no more questions, I'll check the bottom of the chat again. And there's still a question on viral release. I will let you guys read on viral release because I've spoken about it several times. Then maybe in the next class I can pick up. But the idea with viral release is that if the virus is not trapped, if the virus is released by a cell, or it leaves the cell through budding, and within the vicinity of that cell is a lot of sialic acid, then that virus will bind to the sialic acid and it will not be available to bind other cells. But if within the vicinity of that cell, the virus has released neuraminidase to clear the sialic acid around the cell, it means the virus will be free. It will not be agglutinated, it will not be clamping around that infected cell. It will be free to infect other cells because the sialic acid has been cleared. This is something I think you guys need to check because I can see a lot of people asking about it. Maybe you need some time to digest it. But the idea is sialic acid is the receptor. The virus will bind to sialic acid and then enter the cell, but a lot of virus will also bind to sialic acid and be extruded with the mucus. So when the cell is dying because of the virus inside multiplying, the copies of the new virus, when they leave that cell, if there's a lot of sialic acid around the cell, maybe in the mucus, the viruses will be bound by that sialic acid to the mucus, and so the viruses will be inactivated by that binding. But if the viruses, the sialic acid around the cell is cleared by the neuraminidase, then it means the virus will be free to go and bind other cells. So that is why neuraminidase facilitates viral release. A question about antigenic drift and shift. We explained earlier that drifting is due to accumulation of mutations, which occurs over time. Shifting is due to genetic reassortment or mixing of the genetic segments, which leads to a totally new subtype. Classification of the virus is alpha, beta, gamma, and delta, which is genus classification, and the types are alpha, influenza virus A, influenza virus B, influenza A virus, influenza B virus, influenza C virus, and influenza D virus. So A, B, C, D for the virus, and then alpha, beta, gamma, and delta for the genera. Can being reinfected by a completely different strain of influenza cause any syndrome like hemorrhagic shock syndrome? This is a question that was asked earlier. I think you should check that. Hemorrhagic shock syndrome is not common with influenza, so I don't want to discuss it too much, but you can check if somebody has published on the frequency of it. I haven't seen. I don't see any other question here. Okay, so I'll stop there. I think all the questions have now been addressed. If you have issues with understanding some of the concepts that I will share in the slide, you can ask these questions in the next class on Friday. Otherwise, I'll stop there for now and wish you all a nice afternoon. Thank you very much.
Thank you.